from the University of Munich in Germany. Before I forget, I want to remind everyone we're having a reception and dinner for him tonight at my house. I think everybody got the email. Everybody is welcome, kids, families, etc. But if you haven't RSVP'd, please do so that we get an accurate head count for food. Jochen received his PhD from Bielefeld University. I'm probably
that's about right.
Yeah. Which I had never heard of. But then went on to do postdocs. I guess when you were at the Max Planck Institute, that was a postdoc?
That was a postdoc, yeah.
And that's where I first met him. So I did a sabbatical at the Max Planck Institute for Evolutionary Biology in Plön, Germany, in 2007 for just a few months. And he was there as a postdoc. And out of everybody that I met during my time there, I found Jochen to be the most engaging and interesting person to talk to about science. It was really fun for me. And he was very excited about crows and was telling me all about this hybrid zone and what he was going to do with it. And it was all sort of just getting started. And then in 2014, he published this phenomenal paper in Science describing this hybrid zone between two crows where they looked at patterns of genetic variation genome-wide and found just one very small region of differentiation. So strong phenotypic differentiation in an otherwise sort of homogeneous genetic landscape, which I'm sure we'll hear about more today. After leaving the Max Planck Institute, he went to Uppsala University, where he was a lecturer for a number of years. He was, I guess you were there for a total of 8 years. Okay. And then moved to Munich in 2016, where he's currently a full professor and has your own, he's a group leader, is I guess what you call this in Germany. Is that?
Yeah.
So he's a birder, and he loves birds, and we're going to hear about crows today, but he also works on marine mammals and yeast and various other things.
Some frogs.
Some frogs as well. So he's got broad interests in diversification and genomics. I've been eager to get him to come here. I think I first invited you 5 or 6 years ago. And I took advantage of the fact that this year he's on vacation with his entire family, with 3 children in tow, and they've rented one of these giant RV campers, and they've spent the last 4 or 5 weeks driving around to all the interesting, beautiful places in the western United States, including Death Valley and the east side of the Sierras, and they arrived last night, and if you come this afternoon, you'll see this giant RV parked in front of our house. But we're very glad that he's willing to take time out of his vacation to come speak to us. So thank you very much, and please join me in welcoming him.
Yeah, thanks, Michael. So as Michael said, this is an off-duty talk. Don't expect too much. So this talk is going to be about staring at crows for the last decade, more or less, right? But the motivation lies somewhere else, of course, and the motivation is that we, as many of you, I guess, want to understand principles underlying the generation and maintenance of biodiversity. And this has been framed very nicely already 80 years ago, just at the start of the formation of the novel Synthesis by Theodosius Lubchansky, where he says, "Experience seems to show, however, that there is no way toward an understanding of the mechanisms of macroevolutionary changes," that's all those species richness that we observe in nature, "which require time on a geological scale other than through a full comprehension of the microevolutionary processes observable within the span of human lifetime, and often controlled by man's will. For this reason, we're compelled at the present level of knowledge reluctantly to put a sign of equality between mechanisms of macro and microevolution, and proceeding on this assumption to push our investigation as far ahead as this working hypothesis will permit." So we, as many others, have a microevolutionary research program that kind of works on this realm within populations and closely related species, trying to understand how biodiversity is generated. And importantly, when this book was written, this part here, kind of novel synthesis, where we have like mathematical tractable parameters like population size, selection, migration, recombination, mutation, hasn't really been amalgamated into one theoretical framework. It now has. But I think for understanding speciation, it is still important to keep in mind that evolution is more than just the passing down of mutations through a hereditary process. And even, again, Jansky has put this on paper 80 years ago. Mutations and chromosomal changes are constantly rising at a finite rate, presumably in all organisms. But in nature, we do not find a single greatly variable population of living beings, which becomes more and more variable as time goes on. Instead, the organic world is segregated into more than 1 million separate species, each of which possesses its own limited supply of variability, which it does not share with the others. So, if we want to understand speciation, we need to understand more than just the evolutionary process, how allele frequencies change within populations. There's something more that has to happen, and that is reproductive isolation. This concept has been coined by Meyer in the middle of the last century, with the idea that we start from some unit, let's just stick with my picture, that as time passes through, develops into two separate or more lineages. And the trick to understand is what happens here in this gray zone, where there is gene flow, where they're still homogenized, what puts a barrier between those development lineages. And those barriers can come in different flavors, in prezygotic or postzygotic forms of reproductive isolation. And that's the program of many of us trying to understand how those barriers are engraved in the genome. Right? How is this kind of encoded to keep those lineages separate? How do we get at this? Like a few years ago, this was entirely impossible, but now we can sequence, and that's what we do as well, all kinds of organisms and study genetic variation at base pair resolution. And again, the theory for this, for the so-called genome scans, trying to find regions of the genome that separate emerging lineages, is an old concept. It was essentially brought up by Cavalista Sforza and then verbalized by Lilliton and Krakauer in the 70s, where they say the variation in gene frequency among populations or between generations within a population is a result of breeding structure, demography, and selection. The breeding structure should affect all loci and alleles in the same way. If there is significant heterogeneity between loci and their parent inbreeding coefficients, the fixation coefficients, this heterogeneity may be taken as evidence for selection. So this was considered, a few years ago, a great tool, and that reminds me a bit of the traveling through California, kind of the gold rush, when we went to those historic state parks. This is the nugget for evolutionary biology, right? We want to find regions in the genome that are more different than expected by neutral processes between two emergent lineages, the so-called speciation genes of burial loci. The concept goes like this: is two populations are still connected by gene flow at the beginning. This is the genome. If you run along the genome, there may be some regions or some loci that are under some form of different divergent selection pressure between those populations, and as time passes, they may reduce overall gene flow, but they may also kind of expand, so recruit other loci that kind of come into linkage with those initial burial loci, to the point where you get linkage disequilibrium between all those novel mutations conveying reproductive isolation, and then you get totally separated evolutionary lineages. And this should be, according to this genome scan model, be manifest in the landscape of some measure of genetic differentiation, for which we often use FST, with those regions that are really kind of standing out, have a higher differentiation than others, that they are kept different by divergent selection, whereas other regions are still kind of homogenized by gene flow. Of course, this model only works if you have recombination, kind of detaching the history of this region from the history of these other regions. And at some point, the whole level of genetic differentiation should rise up, and if you do this between humans and chimps, for example, it should be up here. And the genomes are kind of, everything is in linkage disequilibrium. And that has been observed in all kinds of organisms. We looked at this in the flycatcher, for example. And you see these gold nuggets of speciation here, which probably have nothing to do with speciation, right? Because, and that is kind of, was known a long time, but has been realized by the community just gradually. There's many papers on this now. There's of course other processes introducing heterogeneity in the in the landscape of genetic differentiation. One process is linked selection, could be linked selection like background selection and purifying selection, or it could be repeated positive selection or meiotic drive, for example, that generate these high levels of differentiation here, but they have nothing to do with reproductive barriers between two species. So, and I'm just introducing this because we look at some genome scans later on in the talk. It's difficult to differentiate between a scenario where just per se evolutionary processes increase differentiation that actually have nothing to do with reproductive isolation from those where divergent selection maintains or increases differentiation against homogenizing gene flow. The pattern looks the same, the underlying processes are very different, and that is a challenge we're all facing right now. There's different ways to address it. You can do sophisticated models if you're able to do so. We're not, so we take a brute force approach, as you will see. Well, we took a brute force approach, as you'll see in the future, like in the next minutes. When I did a review kind of about on 70 or so speciation genomic studies, I realized that this phenomenon really was not well appreciated whatsoever. FST was measured in almost 80% of those studies. Gene flow was even measured in 1/3 or so of them, which is the precondition to differentiate between these two models. Recombination and mutation rate hardly ever measured, and there's only some composite parameters that are reflective of selection somehow that have been measured in only very few studies. So it seems that we do not appreciate kind of the evolutionary process enough, which can generate these kind of patterns and differ very much. We have been looking too much at patterns, not at process. And so what we do in the lab is we look at these kind of early divergent lineages, as Michael said, in a different set of organisms from yeast to killer whale, pretty much organized by body size here. But what I will focus on today is the crows, because that's kind of a research program that's been going on for a decade, where we kind of understand most of the, or some of the underlying principles. So the crows you have here are usually black, right? I've seen the American crows flying around. I actually took a raven for a crow here because the size seems to be much smaller than in Europe. But they look like this pretty much, right? Like the carrion crow in Europe, and they're the sister species. So the American crow is the sister to this whole species complex of the Eurasian crow. And the Eurasian crow, you find that in a leapfrog pattern with black crows here in the west. Interspersed are those grey crows, the hooded crows, and back on the black crow here to the right. And down here, we don't know much about the contact zone if there is ongoing hybridization, but there's another kind of white-collared crow here in Asia. Why is this system interesting? Well, if you look at genus Corvus, there's roughly about 40 or so species. You find a repeated emergence of this color pattern, this pied plumage pattern. And of course, this could be, our brains could be playing tricks on this, but you can order them, the species pairs, kind of along the line of progressive reproductive isolation, and we find this pattern in the Indian house crow, where it's more or less clinal, and that's still one species, they happily interbreed. We find it here in this context that we are studying, where there seems to be some reproductive isolation because we get the formation of those parallel hybrid zones here, one in Europe and one in Asia. We see it between the Darwinian and the Eurasian jackdaw that occasionally hybridize, but they're rather advanced in their speciation process. And then there's these two Indonesian crows, the python crow, and the Corvus unicolor, that are super rare, but they're like good species. So we could imagine that the color trait plays some kind of role for pre-zygotic isolation and the formation of new species, which is supported by the observation that they do mate assortatively according to their plumage phenotype. And birds strongly imprint also on their parents, and the mate choice is influenced by what they see. That's the setup of the system, and the second important observation maybe is that we do find when we look at the hybrids, so this is ancestry coefficients, if you're all black, you're of black ancestry, carrying crow ancestry, you're all gray, you're of hooded ancestry. The hybrids, they show different admixture proportions, which is also what we observe in the field. So they have gene flow across the hybrid zone. There is back crossing. So we have a system where we have very close related lineages with 2 or 3 parallel hybrid zones. So to look at this, we first had to characterize the phenotype more clearly. And if you look into the literature that has been described already in 1928 by Munchen in Germany, the only observable difference really seems to be coloration. They've measured all sorts of things, but there's much more difference, like from the north-south gradient, than there is across the hybrid zone between those species. So Linné called them species, and they're still called species, which we can discuss later whether they are or not. So first we looked at the phenotype, pigmentation, characterized this more closely, and behavior. I'll come to that why. And then kind of complement this with genomic variation. And that's kind of the brute force approach I was talking about. So we try to attack the problem from different angles. So for the phenotype, we collected populations from Spain, Germany, Poland, and Sweden in a 2x2 design. So you have equidistant populations of kind of both origins, carrion and hooded crows, which is not so easy actually because crows live in trees. They live very high up. So that's a crow nest, and that's me. And that's Yammer holding the rope. And so it was very hard. We climbed in vain very often to find the nests empty. Then, my son actually got the idea, why don't we use drones? It works perfectly, so you just fly up. And then we actually at least don't climb in vain anymore. We know the age and the number of individuals, and then we can climb. It was very efficient. So once we had the nestlings, we kind of brought them into a common garden environment twice. Once we did this in Bad Hofzell at the Max Planck Institute for Ornithology. From all those locations, put them in those boxes, flew them into a common garden environment. Very hungry animals, you have to hand raise them every couple of hours. But that's important, of course, if you want to study the phenotype at the level of the gene expression, which is super nice because that lies between the genome and the observable morphological genotype. We need some kind of a common garden environment. And that's the best we can do in crows. Ideally, to have a second generation, but well, it doesn't work. So the gene expression experiment went like this: that we plucked the feathers of them. We're actually lucky that this worked, because by this we could stimulate coordinated regrowth of those follicles, and then measure in those feather follicles gene expression at the time when melanin, so the color pigment, is deposited into the actual feathers. And we have a contrast between the head and the body, which is black in the carrion crow and black and gray in the hooded crow, but also a contrast between species for both body regions. So that's a 2x2 design to try and figure out whether there are differences in genes that actually pattern the body or differences in genes that actually just crank up or reduce the amount of melanin. That's the result, quickly. That's the head, that's the torso. Most of the genes were actually differentially expressed between head and torso in both species, in hooded crows and in carrion crows. And like 400 of those we shared between two species. So we interpret this as kind of there is genes that pre-pattern the body. My colleague Nicolas Rompel looks into this in Drosophila flies. It really seems there's a trans environment on a bird that defines the different patches. And then there's a cis environment, if you wish, that kind of differentiates how much color you put onto this. So I'm pretty sure if you reduce color in a carrion crow, maybe even in the American crow, you get a hooded crow, because the patterning is still there. And the genes we find here, that's Hox genes, that's agouti, so that's the typical candidates that are known for establishing kind of a trans environment of body patterning. What was interesting though is kind of that there was these 23 genes that were differentially expressed between the two bodies, and there was none differentially expressed in the heads. So those, of course, are the primary candidates for generating the phenotype that we're interested in. 23 genes have not been hit by mutations, right? If you look at gene expression, we're always dealing with metabolic networks. putting this into context, and that's of course our luxury that we have all this mouse work, and assume at least that in birds things work rather similarly. So we have the melanogenesis pathway, and if we put those differentially expressed genes on the pathway, we see that there's a lot of red, and those red genes are overexpressed in the black feathers. We of course also did the same for other tissues, and there was hardly any differential expression for the other tissues. So definitely the only phenotypic difference that we seem to find is in the kind of melanogenesis pathway in the feathers. But what's striking though is that everything kind of seems to coalesce onto this gene MITF, which is known to be a major transcription factor regulating melanogenesis. But MITF itself was not differentially expressed. And that was a bit, of a bummer. So it should be really, if you look at this pathway, it wasn't. One explanation could be that MITF is not specifically expressed in the melanocytes, but is just ubiquitously expressed, and if you do RNA-seq, as we did here, the signal just gets diluted. So what we did is we designed probes to measure single cell, like cell-specific expression of the feathers in a histological context. The feather is a fantastic thing, right? It starts from an epidermis, which is two-dimensional, and then kind of goes into this fantastic three-dimensional structure with the rachis and the barbs and the rami. And so we took cross-sections and longitudinal sections of those feather follicles, and we're able to stain the melanocytes, which you see these large green cells here, and see how they develop from the base of the feather further up to kind of older parts of the feather. And then what we could do is using padlock probes, they kind of stick onto the mRNA molecules with a rolling circle amplification. You can visualize single mRNA molecules. And that's kind of this picture here where you see one white dot here would be a single mRNA molecule. We do this for a set of kind of candidate genes that we're interested in, and then we can measure the specificity of those genes overlapping with melanocytes or not, right? Then we get a measure of melanocyte specificity, and we see that MITF indeed is not very specific. It's rather ubiquitous. So this is actin, which is entirely ubiquitously expressed. These are TER1 and other melanogenesis genes that are only expressed in melanocytes, and MITF is somewhere in the middle. And if we subtract, if we just look at expression in the melanocytes, MITF is also differentially expressed. So we're back to this pathway, it all makes sense, right? And just by looking at gene expression, we can now define certain genes that may be kind of causal to kind of generating this differential expression downstream in all this pathway. There's one, this HPGDS, which is upstream here of SOX9. There's the MC1R route. There's the MAPK route through this gene, which is influenced by calcium channels, which are not shown here. And then there's this NDP gene, which turns out to be super interesting because it was differentially expressed between gray and black, but also was different between the shaping, right, between head and body. For the two species, there was an interaction. That was just the color side. So gene expression suggested that there was no difference in other tissues, pretty much, except for some gonads, for example, which we haven't really followed up. So there has been the observation that melanic animals quite often also tend to have differences in their aggression behavior, for example. So there could be a tentative pleiotropic link between color, gene expression leading to color differences, and behavioral differences. And in crows, there's a paper by Saino and Scatizzi in 2005 where they observed different levels of aggressiveness between the carrion and the hooded crows, which might be a reason why, and that's work by Haas and Brodin, why the hybrid zone has been observed to shift in favor of the black ones. Right? So reproductive isolation could be helped, or it could be at least one component that they have different social dominance behavior. And the mechanism for this has been suggested by a group of Alexandre Moulin. There is a potential link through the POMC gene, very upstream of the kind of melanogenesis pathway, that links the hormonal pathway to melanogenesis. So we investigated behavior. We brought single-sex groups of crows into aviaries, measured affiliative and aggressive interactions. It's shown a network of aggressive interactions that would show that these social dominance hierarchies here, measured by an ELO score, which is a measure from chess pretty much, that they're stable across time and that there's a clear social dominance hierarchy. We then repeated this with dyadic encounters of carrion and hooded crows to see whether carrion crows are dominant over hooded crows or the other way around. But what we found was pretty depressing. There's just nothing, right? They vary a lot in size. Whatever you do, they just show no behavioral differences whatsoever. The only difference we observe is pretty much a sex effect. That supports kind of the gene expression experiments. We also had a kind of a look at the pituitary, which governs this. There doesn't seem to be any differences other than color. So this is a system where we would predict that color really drives the maintenance of this hybrid zone and kind of maybe the first steps to speciation, whether that's the way you want to think about it. So that's why we did a genome assembly. Now we're running in version 5, right? Those methods change all the time. We started with Illumina, then was PegBio, then there's optical mapping, then there's Hi-C. It's a horror for the lab because we have like all these versions flying around and everybody works in different versions, but that's the way it is. That was the first version, and we kind of did a genome scan. We kind of characterized the genomes of 15 individuals from each population again. And what you see here is a PCA from this genomic sequencing based on 8 billion or so SNPs. And what we see is that the German individuals here, shown by those black dots, cluster very closely with the Swedish and the Polish hooded crows. So genome-wide, Germans are hooded crows, right? So these black genomes are gray, whereas the Spanish genomes are black, if you wish. So the Germans are much more different from the Spanish than from the wrong species, if you wish, genome-wide. And that's also kind of shown here in this admixture ancestry plot. If we now run across the genome, and that's Michael's introduced that already, and compare, for example, two hooded crows, we see that across all chromosomes, there's hardly any differentiation. Right? They're the same. They look like one panmictic population. If we do the same for Spain versus Germany, they have a much higher level of differentiation. So they have some history of being separated. If we then compare any black versus any gray, and here this is illustrated for Germany versus Sweden, we see one major peak of differentiation here on chromosome 18, which is, and that's important, not visible in the black-black comparisons, right? So that seems to be unique to black and gray. So we got our speciation island, we thought, right? And so in total there are 83 fixed differences between carrion and hooded crows across the hybrid zone, Germany versus Poland, or Germany versus Sweden. Out of all those SNPs, 81 sit in this region. So we're really afraid that this is just some kind of very weird artifact. But whatever we tried, it doesn't seem to be an artifact. Let's zoom into this peak, and we find those 3 genes that are hard to differentiate because this region doesn't seem to recombine. It could be an inversion, but we haven't pinned it down yet. We'll talk about this later. So there's the calcium channels. There's this PRKCA gene and AXN2. They're all known to influence color. So they sit here in this peak. And interestingly, that was very hypothetical, of course, very speculative. There's another gene, this RGS9, sitting here, which influences contrast vision, which could be kind of the coupling of a mating trait and the preference trait gene. But this is, of course, don't trust it at all. This is just a speculation. But like this, it would be a textbook example of how you couple in an inversion a mating trait and a mating preference, and then kind of set off the speciation process. Instead of this gene, imprinting would actually also work. So we do not need a gene for this. We just need a trait. Next, this was all the European hybrid zone. So next, we proceeded to the Russian and the kind of this Asian zone and asked whether it's the same process. First of all, we see that this kind of Asian crow is just a drift tree, arose independently from the carrion crow, from the hooded crows, and they're also all very, very young and have hardly any genetic variation. So we estimate that they separated about only 10,000 years ago, so at the onset of the last ice ages. When we do something called a PSMC analysis, including coalescent, we estimate that all crow populations, these are representatives of all those populations, kind of have a common ancestor here like 350,000 years ago or so. So we really see that population differentiation only at this time point, which is of course very shaky, but it's a young system. Right? You can see that all of crow radiation in Eurasia is still young. And when we do this as a control, we compare same colored populations with increasing levels of FST, increasing levels of genetic differentiation. What we see is that these speciation peaks pop up everywhere. Right? So there's clearly some common peaks that show up here with chromosome 1, chromosome 2, and it's about 1 to 3 or so per chromosome. And that's what I said before, right? This is probably due to other processes that have nothing to do with gene flow or resistance to gene flow. That's some form of linked selection that could be centromeres, for example. But if we then try to subtract those, very brute force again, this is kind of these peaks here, the peak remains for Europe. The peak for the Russian zone is very, very shallow, and hardly anything remains if you subtract those common peaks. And in Asia, we get a very strong peak as well, but they all sit on different chromosomes. So they don't show the same genetic regions. If you look into them, of course, you find the same, genes that are shared in the same width part of this kind of melanogenesis pathway. So parallelism may be on the level of the pathway and not on the level of the individual genes. Looking at this prompted us to look into the genomic context more, because we don't even know where the chromosomes are, or where the centromeres are in crows. And what could well be is that our peak is very close to a centromere, and that the color has not been selected in its own right, but is just linked to something, for example, centromeric drive element. So we tried to get at this a bit better in crows, doing PacBio assemblies, and we found those repetitive anchored maps, that's repeats, mini satellite repeats, that co-align very often with regions of reduced recombination, regions of increased differentiation and reduced nucleotide diversity. So it seems that those repetitive regions are possibly in heterochromatic regions of the genome, and that influences our population genetic parameters. And that is definitely something we should take into account in the future, not treating a chromosome as just one line, a linear molecule of with the same properties, but there's clearly heterogeneity in the properties. And so what we really want to know is the recombination rate across the genomes, because in centromeres it's supposedly lower. But that's very difficult in crows, because we cannot make large pedigrees in crows, doesn't work. So what we tried is to estimate the recombination rate from single sperm cells. If you have one individual, a male individual with the maternal and paternal chromosome, it will recombine for each sperm cell. And in principle, if we sequence single sperms, we should be able to detect those recombination breakpoints. It works, but it's difficult. So we've tried that in zebrafinch now to try and address the question that was the whole goal, right? To see whether there is an inversion in crows or not, to see whether recombination rate is really reduced in the inversion breakpoints. And we used the zebrafinch as a model for this, where we know that we have individuals that are heterozygous for inversions and individuals that are homozygous for the inversion. But the first result, by looking at this kind of sperm-based male recombination estimate, is that the estimates for recombination rate, shown here inside of the circle, correspond rather well to the estimates that are based on LD. So the method seems to work. In terms of inversions, I think we still have a lot of technical issues to solve. We do not see right now a clear pattern that inversion breakpoints have reduced recombination. But that's technically still not fully resolved. Okay, so does one peak explain everything on chromosome 18? This is back to the European crow hybrid zone. Probably not, because if we look at the hybrids, they have different patterns from a color growing always kind of tighter, or to the whole body growing a bit darker. So there seems to be at least more than one locus involved. And to investigate this, we made use of admixture in the actual hybrid individuals. So nature did an experiment for us. They mixed the crow genomes in the hybrid zone, so we can kind of do admixture mapping to try and really get at the genetic basis of coloration. And that is actually interesting because when we submitted this paper for review, which I'm going to show you in a minute, the reviewers said, well, it has been shown that chromosome 18 is the region for the color difference, and it hasn't been, right? It's just FST. So we never associated this genetic region with the phenotype, really. This is overgeneralization. We just took two populations, some were gray and some were black, but that doesn't necessarily mean that this region is associated with gray or black, right? It could be a historical coincidence. So that's why these hybrids, these admixed hybrids, are very, very valuable in addressing this question, really linking the phenotype to the genotype. So we did a Transac in Germany here, and one in Italy, with the help of Nicolas Sainon, and measured different patches here along the dorsal and ventral side of the crows, very simply saying light gray, dark gray, or black. Inter-observer reliability was very high for this simple way of measuring. And if you do a correlation between all those patches, the correlation is really high, showing that it is governed by not many genes. It's rather, if you're black here, you're very likely to be black here too. But it's not perfect, of course, because there seems to be another element disassociating those different patches. The next step was then to summarize this phenotypic variation in one principal component axis, which explained, get me right, about 80% or so of the variation. So it explained a lot of the variation, and that's this first axis. And if you have high values here, you're black. If you have low values, you're gray. You're a hooded crow, but in between you're some form of a hybrid. And this is just the frequency of individuals in the hybrid population. If we then do a genome-wide association mapping on this, what turns up is chromosome 18. I was really lucky to see this because I was still afraid that this whole chromosome 18 business was a totally artifact with whatever. It didn't seem to be because it was really associated to color. What also showed up is this NDP gene, which was highlighted already by the gene expression analyses, and an LRP6 gene, which was one of the genes that was commonly found between the Russian and the European hybrid zone. So it made astonishingly much sense. Altogether, this G was explaining almost 90% of the variance. So it really seemed to be there's only a few genes involved, major effect genes, the biggest one sitting on chromosome 18. The problem with chromosome 18 is, as I've shown you, there's several genes that are candidates for modifying the level of an element. If you look at the hybrids, we just don't see recombination there. It's very, very rarely not. So these are double dark carrion haplotypes, and that's for the 2 megabase region or so. These are the double light genotypes, so all hooded crows look like this, and that's the hybrids. And this could be kind of recombination here where it goes from heterozygous to homozygous, here double recombinant. There's one other recombinant, but in general there's just no recombination in this region, which could confirm again that it's an inversion, or it's close to a centromere, or we don't know. So if we now model coloration as a function of those genes, the best model emerges. The best model that emerges is the result of epistasis between the factor on chromosome 18 and this NDP gene. So that's how it goes. And it's recessive epistasis, so if you are double dark on chromosome 18, you're black, regardless of what you have for the NDP gene. You're black. If you're double light, you're not necessarily light. So you can be a pure hooded crow, but you can also be pretty dark. So the NDP genotype modulates the color unless you're double dark for chromosome 18. And those two genes explain most of the variation in coloration. That's just the phenotypic aspect. If we then run genomic clan analysis or geographic clan analysis across the hybrid zone, that's easier to explain here. We see that genome-wide, the collide is very, very large. It's about 400 or so kilometers. So genome-wide, the hybrid zone is 400 kilometers wide. Whereas for those chromosome 18 and NDP, those genes, it's very narrow, about 50 kilometers or so. And that is an indication that it's not only just governing color, but it's on a clearly divergent selection that prevents the allelic variants to cross the hybrid zone boundaries. And the same is pretty much shown if you do not use geographic space as the x-axis, but admixture space, kind of ancestry coefficient of the whole genome. Again, chromosome 18 and NDP show up. And that's an important finding because it demonstrates that these two genes not only govern pigmentation, but that they're also under divergent selection preventing the allelic variants to cross the hybrid zone. And if you look at the summary slide from Hudson and Price, there is a clear relationship between the width of the hybrid zone and the age of the hybridizing taxa. So usually if you're young, you have very, very narrow hybrid zones. When you get older, the other way around, when you're young, you have very wide hybrid zones because they're kind of still, they merge. When the hybridizing taxa are older, the hybrid zones get much, much narrower. Seems to be strong selection. The crow is a clear outlier. The crow is very young, but it has a very narrow hybrid zone. And that was a puzzle for long, but if we kind of factor in those genomic insights now, well, the hybrid zone is really only narrow for those two genes, but it's super wide for the rest of the genome, right? So reproductive isolation here, determining this is all morphologically based, of course, the hybrid zone width, determining that the morphological hybrid zone is merely governed by those two genes. Reproductive isolation only relates to those two genes, not to the rest of the genome, which still floats freely. A few more minutes, 2, 3. Yeah. We then did PacBio sequencing on 31 genomes, including several species, and I apologize for this slide. This was a holiday slide, I just stuck it in. So you don't see those crows, but what's important here is these two crows. That's our carrying crow, that's the hooded crow again. Like all the ancestors here have what is that now? Like, don't have the insertion here. But then when at this lineage, when the Eurasian crow species complex, this is where you get kind of homozygote and heterozygotes with an inserted LTR retrotransposon in this region, upstream of NDP, which is entirely fixed in the hooded crows. So what we believe is that this is a regulatory insertion deletion mutation, kind of that governs the expression of this gene that then kind of contributes to the coloration. And just as we wrote this up, we found that Vickery et al., so that's Mark Shapiro's lab, they found in pigeons, pretty much exactly in the same region, the copy number variation governs color patterning in those pigeons. It wasn't that NDP was not known as a color gene before, I think. So that kind of lends support to the idea that NDP really drives that, and that might be a causal mutation. It's difficult to test in crows, but we see that the heterozygous and the homozygous ancestral ones have higher expression in this gene, making them more black than the derived hooded type. Okay. Last idea. So maybe we're actually faced with just speciation by perception. We just have a couple of genes, or 3, modulating a trait that's perceived relevant for mating. And if you imprint on this trait, and that's been modeled with a neural network by Rodin and Haas, and kind of mate accordingly to your parents, you may get a very narrow hybrid zone which reflects the actual pattern. And we revisited this with a more complicated and explicit model now, putting in our NDP and our chromosome 18 and the epistatic interaction, having a matrix of mating probabilities, suggesting that the crows that are more similar in color have a high mating probability. So if you're the same, you have a mating probability of 1. If you're very different, you have a lower mating probability. And we stick this kind of matrix into a model where we estimate those numbers pretty much. can tell you more about the model, but time doesn't allow. And then we use this data, the clines that we observe, the allele frequencies of chromosome 18 and NDP across the hybrid zone to kind of estimate those parameters. That's the data. That's what the model predicts after estimating it, so they're pretty close. And that's what it looks like. So just to finish here, I'll run 10,000 years of evolution in this model. We start with black crows in the west. It's a model of secondary contact, which is suggestive for this kind of system. And gray crows in the east. They meet here at point 0. And then they start mating according to those mating preferences based on color. And the genotype is always explicit in there. Right? When they mate, they get offspring, and the genotypes follow Mendelian ratios. There's 3 loci. This is chromosome 18, NDP, and a neutral locus. And this is just the 3 neutral loci. Okay, we start. I stop. So 180 generations, that's about 1,000 years or so. We see that the neutral locus kind of starts moving rapidly across the hybrid zone, whereas chromosome 18 remains very steep, NDP as well, but it's moving eastwards. So the carrying crows win. Carrying crows kind of outcompete the hooded crows. The neutral locus seems to be kind of almost swamped. And then something really strange happens. Now the hooded crows start winning. And if you run this, this was 10,000 years, so this is what we think was more or less the time where we are right now. And if we project this into the future, the hooded crows outrun the carrying crows, and now they're probably in the Atlantic Ocean somewhere in France, and they're gone. Right? So the prediction from this model would be that the hybrid zone is absolutely not stable, it's very dynamic, and depending a bit on how we set up the architecture, if we do an additive architecture, the outcome will be different. These dynamics are highly dependent on the genetic architecture and on the mating matrix. Okay, so what we need is ancient DNA. If there's a student who wants to work on ancient DNA, come to me. We have a lot of bones, human 8 crows in the Stone Ages. We have a lot of samples actually, so they just throw them away. We have sampled that goes back to 73,000 years, and that's a project we started, but then many things came in the way. It works, we get DNA out of those, so what we now want to do is to just retrace the history through time in Europe to see what our model predicts makes sense. With this I would like to thank you. Thank all the people that worked on this. Julia and Benedict worked on the behavior. Grzegorz and Jelmer did much of the genomic work. Matthias on the structural variants with the pectoral data, Chichi the single cell expression, and Oli and Josh worked on the zebrafinch recombination, and Dirk was mostly responsible, colleague mostly responsible for the actual modeling. Okay, so I hope I convinced you that crows are not just for shooting, they may give us some insights into evolutionary biology. Thank you.
We'll let people who need to run out to a class escape, and I'm sure Jochen would be happy to take questions.
Hi. So you mentioned that there's sortative mating, or evidence of it, and you have high FST between these different morphological types. So I'm wondering if potentially you could have a situation where you have ecotypic differentiation after speciation, and then a sortative mating that happens after that feeds back on a hybrid zone. And so I guess what I'm saying is, if you look at alternative alleles at these loci that you see that have high FST between these 2 morphs, if you age those alternative alleles, do they date back to as old as at least the speciation event or older?
The dating is tricky, and we've never really addressed this because right now the working hypothesis is that we had. It's so difficult to reconstruct the history of this, because if you look at the drift tree here at some point, the German population looks very much like the hooded crow population, right? And that is probably because of secondary contact that they met, and then kind of the carrying crow genome was swamped by the hooded crow genome. But we do not see, like, all the hooded crows look the same from all the way from Russia to Europe. And I think we lack one population that could be down in Iran or Iraq that may be actually very differentiated. And if we got that, we could age the whole genome and see whether the part of chromosome 18 belongs to that or not. Otherwise, they're not much more. I mean, there's only 81 fixed differences, right? They're not very old, and that depends on the demography, if they've gone through bottlenecks or not. So whether the fixation is due to novel mutations or due to existing variations. So I think we may be tricked by the sampling if we try to estimate the age of those. And ecology, I don't think, plays a role at all.
Or even differences. So I guess that would be under like the assumption that in this population it's ancestrally segregating polymorphisms become fixed between species, or maybe a hybridization event introduced some new
I mean, for this NDP gene, definitely happened. This happened. But it was segregating ancestrally in both populations, and then it was kind of, that's what we think, was taken by the hooded crow to make it prettier, if you wish, right? If there was so if you're in a heterozygous state on chromosome 18, you're rather hoodie-like, but still pretty ugly. And then if you put those, kind of, if you link those epistatically with this NDP variant, then you get bright and really kind of yellow. Why this is then selected for is really difficult to say, right? It might exploit just the visual mechanisms of crows that they like it for some reason. This bit is so difficult to explain why this mutation arose in the first place and why it kind of took off. But it can't be ecology, really. Maybe in the ancestral population somewhere. Hard to trace.
Can you cross-foster these and get them to imprint on the wrong phenotype?
That was the idea, but it doesn't work because, I mean, you actually thought about transferring them across the hybrid zone, but that's impossible, right? 80% die in the first year. Pretty much you have to wait until they're socially dominant enough to build a territory, which takes 10 years or so. And by then you won't have any tagged.
But what if you just take eggs and put them with a parent that looks different from...
Yeah, but you have to wait what kind of choice they make 10 years later, right?
That's 10 years. You're young.
And then when we tried behavioral tests, we built Skinner boxes around them and presented them with kind of different, shades of gray and contrast, but they didn't cooperate. They were bored. They tricked us there. So getting at the actual mechanism this is just where we're working on use. Right? This is where we hit the wall, I think.
The other question I had is somewhat related. So, the initial mutation must be disfavored. If they're...
It's frequency dependent, yes.
Exactly.
Yeah, that's what we just don't
So
understand.
how does this become established in the first place?
We don't know. It doesn't make sense. It could be drift, right? It could just be that it's at a very low frequency. Because it doesn't seem to be favored overall. It's also what the model suggests, right? It's frequency dependent. If you're the odd one out, you're not preferred. So there must be some kind of population bottleneck at some point. That's the best explanation I have, but it's hard to imagine. Or this is pleiotropically linked to something else which we just don't have any idea of.
But you've only got this one small genomic window.
Just curious, is there multiple paternity in any nest? Have you looked at that to see how strict monogamy is?
There is, but in a very, very small proportion. So I think there was 3 nests or 4 in Germany out of those, was that 50 or so that have extra paternity, and those were excluded for the model, right, when we fit the model. So it's very low, and we actually tested the idea, and that was why we did that in the first place, is to see whether, if you mate it with the wrong species, so to say, or subspecies or taxa, whatever you want, whether you then kind of shop the right genes through extra paternity. But we don't have no evidence for that. Yeah. But that would be super nice, right? But they use extra paternity to compensate for the social error that they committed, choosing the wrong partner.
So, wondering if you had some thoughts about the whole islands of divergence and how this relates to that. Like, do you think this reproductive isolation would spread in the system, or is there just so much gene flow background that it's just a stable epistasis and it's probably going to be?
That's hard to say. But it conveys some degree of reproductive isolation for the whole genome, not right. And the simulation, if you believe that, shows that very clearly. Genome-wide, there's soon going to be homogenized, or one color goes extinct if it doesn't stay stable over a long time. But of course, if you have bouts of if ISHs happen again, they might end up in the refugia again, and then you may accumulate other differences that come into linkage. But I do think you need almost some kind of allopatric separation there.
Just a comment about pleiotropy. If the LTR retranspose on insertion is indeed acting as an enhancer, they tend to act as enhancers in hypomethylated regions. And so you can imagine that a stem cell type situation of, like an early feather growth would be a hypomethylated tissue. But as well, other hypomethylated tissues where you have activation of that enhancer would be likely the sperm or the eggs. So those could be candidates for where you would see pleiotropy playing a role. I don't know if you can basically do some gene expression tests in sperm and eggs, but that seems like a potential area.
What we just discussed that with Amin, right? The other tissue where we do see interesting differential expression is kind of the gametes. So, and we don't know whether this PRKCA gene, for example, might be linked, so that there might be a link to some post-zygotic reproductive isolation as well, which we haven't really figured out yet. So there's no evidence from the field that there's strong post-zygotic isolation, but there might be, at least if you look at the gene expression data. Yeah, makes sense.
Other questions? All right, if you want to chat more with Jochen, come to my house this evening, and let's thank him for a great talk.